Well, thank you very much to Ms. Hirschberg and Dr. Go for inviting me here, and also for helping fund my research. I'm going to hopefully convince you today that my proposal is worthwhile, and it uses a genetically engineered mouse model to evaluate the role of macrophages in pancreatic cancer formation. When I was a second-year medical student, and that's the year when we learned about diseases, I kept a running list of what diseases I really didn't want to get. And nobody wants any diseases, of course, but at the top of that list was pancreatic cancer for me. And that was 15 years ago. And I think if there's a second-year medical student here at UCLA or anywhere who's keeping that same kind of list, it might still be on the list because it's such a difficult disease. This is a statistic, or a graph showing how difficult this is. Starting in 1975, this is from the SEER database from the NCI. You can see the mortality rate for pancreatic cancer has basically been level, with even a hint of an increase at the end, despite many new types of drugs and other things coming online. I'll skip past that. This is in comparison to a lot of other kinds of tumors. At the top is lung cancer over the same time period. Here's colon cancer. Here's pancreatic cancer again. And then leukemia and gastric cancer. And I could have chosen other cancers as well. But you can see one of the differences is that these other cancers have had a decrease in their mortality over that time period. And so why might that be so? There are many reasons why that might be so. One of them is prevention. You can imagine that for lung cancer, decrease in smoking that's been taking place has led, at least in part, to the decrease in lung cancer since the mid-1990s. Another is early detection. We can take as an example colon cancer and the fact that colonoscopies, regular colonoscopies, can find the cancer before it spreads and lead to a cure for this colon cancer. Another way that the mortality can decrease as well is through the advent of new drugs, new ways of treating these cancers. And I'd like to compare pancreatic cancer with colon cancer. I'm a GI oncologist, and so I treat patients with both. And for pancreatic cancer, and I'm being a little simplistic here, there's basically, once you get to the advanced stage, there's one drug to use, and that's gemcitabine. Now, I said I was simplistic. There are some other drugs that can help a little bit. The small molecule inhibitor, erlotinib, can add a couple of weeks, but it's expensive. And there was a study out last year in the New England Journal that showed that combination therapy can be helpful. In my experience, it's very toxic, and for the majority of patients that I treat at least, gemcitabine is the mainstay of therapy. If we compare that to colon cancer, there are a lot of different drugs that we can end up using, and a lot of these have come online in the past decade to 15 years. And it's led to a decrease, or actually an increase in survival. Back when 5-fluorouracil was the only way to treat colon cancer, metastatic colon cancer had an average survival of about 12 months. Now it's over 30 months. With the advent and use of all of these different kinds of drugs. So at least in part, one of the reasons that the cancers that I showed before have decreased in mortality is because of new therapies. And that's not the case for pancreatic cancer, unfortunately. So why is that? Why is pancreatic cancer different than other solid tumors regarding resistance to available therapeutics? Those other drugs are available, but they don't work very well in pancreatic cancer. So a simple way is just of looking at this is to look at what the differences are in pancreatic cancer compared to other solid tumors. I'll pick colon cancer again. This is a picture of a colon cancer, pretty standard colon cancer. It shows sheets of cancer cells. There are some glands forming. This is an adenocarcinoma. There are other cells interspersed as well, but the majority of the tumor is comprised of cancer cells. In comparison, pancreatic cancer can be very different, and often is very different. There are cancer cells, and you can see them with these open arrows, nests of cancer cells throughout the tumor, but the vast majority of the tumor is not comprised of cancer cells. It's comprised of stroma, including, as you can see by the black arrows here, fibrosis, as well as inflammatory cell infiltrates, indicated by the white arrow. So there's something very different about a cancer, a pancreatic cancer, from a histologic standpoint. There is evidence that the stroma can play a role in resistance to chemotherapy. And I'm going to tell you talk a little bit about a paper that was published by Olive and colleagues, and they used a mouse, which, for the sake of brevity, they and I will call a KPC mouse. And this is a mouse model that develops pancreatic cancer. It's similar to the mouse model that was mentioned a little bit ago. Let me take and step back just a little bit and talk about this model, in part because it's a mouse model that we use in our laboratory often, and it's part of my proposal. Pancreatic cancer forms in a stepwise manner. You start with normal pancreatic tissue, and then it progresses through a series of pancreatic interepithelial neoplasias, varying degrees, 1, 2, and 3, getting more aberrant as they go, and then finally to frank adenocarcinoma, where it breaks through the basement membrane and starts to invade. And work by others has shown that there are genes that are dysregulated along the way, and they correlate to how far along you are in this tumorigenesis process. Hingorani and colleagues focused on a couple of genes. Obviously, KRAS is important for pancreatic cancer. It's mutated in 90% of pancreatic cancers, an activating mutation. And then they also focused on P53, which is a tumor suppressor gene, common in many, many cancers, all cancers almost, and altered in 70% of pancreatic cancers. And what they did is they developed a mouse that conditionally expresses an activated form of KRAS called G12D, so that it's constitutively active, it signals no matter what, and an altered P53 that has the mutation R172 to H. And this mimics the loss of heterozygosity that is found in pancreatic cancer. Using the CRELOX system and using a PDX driver, which is expressed in early pancreatic formation, the stop sites can be removed from in front of these genes. The mice express these aberrant genes, and they develop cancer, pancreatic cancer specifically. And this pancreatic cancer mimics the human counterpart in many different ways. It looks similar. So histologically, it has areas of tumor cells, lots of fibrosis, lots of stroma. It also mimics it clinically. These mice develop frank adenocarcinomas of the pancreas. It metastasizes, and it eventually leads to their death. They also develop ascites and other clinical conditions as well, and they die usually around 5 months. The last reason to use these mice is that they've been used in many different experiments, including experiments to look at therapeutics to see whether they work. So the Olive group took this mouse, this KPC mouse, and very simply asked, are they sensitive to gemcitabine? Many patients will not be sensitive to gemcitabine. The majority, in fact, will not. And then they assessed the tumor growth. And what they found, and this column is the controls, and these are 2 different doses of gemcitabine, that for the majority of mice, there was no sensitivity to gemcitabine. There were 2 mice down here shown in yellow that were transiently sensitive to gemcitabine, but then they were not after that. In comparison, they then did an experiment which looked at tumors grown as xenografts. So injected either orthotopically into the pancreas or underneath the skin. And the difference, one of the major differences is with regards to stroma. These tumors form, and they're primarily tumor cells. There's intact vasculature. The microenvironment is very, very different. So they took tumor cells from a tumor, which they resected from one of these mice, grew them for a short period in tissue culture, and then injected them subcutaneously into mice. And then did the same experiment. Looked at gemcitabine versus the vehicle control. And what they found, they also did this as well with some human pancreatic cancer cell lines that they injected. What they found with this experiment, which is shown here on the right, that 3 different cell lines all were sensitive to gemcitabine. They also performed it with a couple of human pancreatic cancer cell lines from patients that were known to be resistant to gemcitabine and saw the same thing. So there was a marked difference. These tumors were grown in a different way. They were sensitive to gemcitabine, whereas if the tumor developed de novo in this mouse, it was not sensitive. Now there's many reasons why this could be so, but one of them is that the stroma is different. And this is the last I'll mention of this study, but the final experiment that they did, they took this mouse, this KPC mouse, treated it with a smoothened inhibitor. A smoothened is a component of the hedgehog signaling pathway and inhibits the signaling, and the hedgehog pathway has been found to be important for the development of stroma. So they divided the mice and either treated them with control and a smoothened inhibitor, and then at the same time, not in sequence here, but concurrently, treated with either control or gemcitabine in each of those arms. And what they showed in the purple was that the combination of the hedgehog inhibitor and gemcitabine significantly increased survival. Now, it was a transient effect, but it was significant. And when they looked at the stroma, there was a marked difference in the stroma. There was much less fibrosis, and the vasculature was much more intact, indicating or suggesting that altering the stroma can have an effect on response to therapy. And so that is one of the reasons why I'm focusing on this. There are many components of pancreatic ductal adenocarcinoma stroma. There are macrophages, lymphocytes, endothelial cells, fibroblasts. I'm going to focus, my proposal focuses on macrophages, and I'll talk a little bit in the next few slides why, and hopefully convince you of that. This is a photomicrograph of a human pancreatic cancer, and stained in brown are macrophages. And so you can see that in most pancreatic cancers, this is an example of one, there's an infiltration of macrophages, often profound. So the first hurdle is leapt over, that macrophages are present. When they are present in tumors, they're called tumor-associated macrophages. So let's take a step back briefly and talk a little bit about macrophages. In general, macrophages can be divided into 2 broad groups that have very different and distinct functions. They can be divided into M1 or M2. And M1 are the classic macrophage functions that we think of, that we learned about in school. They're involved in immune surveillance, phagocytosis. They do antigen presentation to other cells, and by doing so lead to resistance to infection. They also produce reactive oxygen species at high levels to kill invading organisms as well. And so this is the classic macrophage function that we think of. In contrast, M2 macrophages are primarily involved in tissue remodeling and wound repair, and they have a very different function. They scavenge debris, they promote angiogenesis, and other processes that are important for wound repair and tissue remodeling, and they decrease immune system activation. And it's been shown that tumor cells can co-opt many of these functions. You can imagine that angiogenesis, decreasing the immune system would be important if you were a tumor and wanted to grow. Tumors are also known as wounds that never heal, so it makes sense that, that these processes would be co-opted by tumor cells. So tumor-associated macrophages are these M2 type of macrophages. They interact with tumor cells. They release cytokines that they can interact with tumor cells. They also alter the stroma through the release of cytokines, growth factors, also proteases, which can alter the stroma. And then there are a number of studies that show that in tumors that have a strong infiltration of tumor-associated macrophages, the patients have a worse prognosis. This is true in pancreatic cancer as well. So it does, macrophages pass the look test as to why they might be important for pancreatic cancer. These are experiments, and it's very light, the number of experiments we've done so far, but these are the KPC mouse in our hands. Actually, this is a normal black 6 mouse, stained for macrophages, and you can see there's a little nest of macrophages here, and a couple in the islet here, but for the most part there are no macrophages, or very few in normal pancreas. Contrast this to a tumor, which is formed in one of the KPC mice in our lab, and you can see, again, profound infiltration of macrophages. This has also been shown by others in other labs as well. So it would appear that this model, the KPC mouse, would be a reasonable model in which to study macrophages. So how can we study the role of macrophages in pancreatic cancer? And what I'm going to propose is a broad brush stroke basically to take a look. Lots of times, if we want to examine the function of a gene in some process, we knock it down. And then we see how the phenotype changes. There's a way that we can do a similar thing with macrophages as well. We can deplete them, or ablate them, it's called. And if we can deplete them and then see how the phenotype changes, this could give us hints as to the importance of macrophages in pancreatic cancer formation. So there have been a number of ways in which macrophages have been depleted. There was a study back in '01 that used CSF1 mice, or deleted mice, null mice. CSF1 is macrophage colony stimulating factor, and these mice have very low levels of macrophages. These mice were crossed with a strain of mouse that's prone to get breast cancer, and they showed that there was decreased breast cancer growth, not decreased incidence of breast cancer in these mice, decreased growth and decreased metastasis. So this is one way. Another way is through a drug. There's a drug called clodronate, which is a type of drug called a bisphosphonate that's used in bone diseases. For example, osteoporosis or tumor metastasis to bones. And in its liposomal form, it's been shown to decrease macrophages. Macrophages are related to osteoclasts, which are important for bone metabolism. And so there's some cross-reactivity. And there's been an experiment that showed that for teratocarcinoma or rhabdomyosarcoma, grown as xenografts in mice, the administration of liposomal clodronate decreased their growth. So this is another way of doing it. And finally, there's another mouse, which was developed 5 or 6 years ago, which is called the CD11B DTR mouse. And I'll explain a little bit more about this in the next slide, but it shows decreased growth of colon cancer xenografts. Let me talk a little bit more about this. So the disease diphtheria relies on a toxin produced by the microorganism called diphtheria toxin. And diphtheria toxin affects cells by inhibiting a protein called EF2, which is important for protein translation. And so when toxin encounters a cell that has a receptor, as most cells do, translation grinds to a halt and the cell dies. For mice, they have diphtheria toxin receptors, but they are not sensitive to human diphtheria toxin. So you can give a mouse human diphtheria toxin, they're unaffected. No cells die because their receptor does not bind human diphtheria toxin. However, if you could put in a receptor for diphtheria toxin, and here it's a simian diphtheria toxin, which binds human diphtheria toxin. If you could put that in certain cells in the mouse and then administer human diphtheria toxin, you could selectively kill the cells which carry that toxin receptor. And so that's what's done. CD11B is expressed in cells of myeloid origin, including monocytes and macrophages. as well as granulocytes. And so what Duffield and colleagues did was they hooked the promoter for CD11B to the simian diphtheria toxin receptor and expressed it as a transgene in a mouse. They then administered human diphtheria toxin and then evaluated the mice and found that the vast majority of macrophages were depleted in the mouse. The other cells of myeloid origin, for example, granulocytes, were for the most part unaffected. So this is a powerful way of inhibiting or ablating macrophages as well. Another important aspect of this is that it primarily depletes macrophages in tumors compared to other glands where macrophages are found. So taking a look at these three ways of inhibiting macrophages or ablating macrophages, I decided to go with the last one. CSF1, the receptor for that is also found on various cancer cells. So it's hard to tell whether the decrease in the growth and metastasis of that is due to a decrease in tumor cells receiving the signal. Liposomal cladronate has off-target effects as well. I mentioned that it inhibits osteoclasts. Whereas CD11B DTR mice, as I mentioned before, it primarily inhibits macrophages and tumors. This is because CD11B is found on those M2 type of macrophages compared to the M1 macrophages. So it would appear to be a nice system for this. So very simplistically, this is not the actual crosses that I will do. We'll take the KPC mouse and we'll cross it with the CD11B DTR mouse. We have both of these in our lab right now through various crosses and end up with a mouse that you can see here that develops pancreatic cancer, that recapitulates human disease, but also in which you're able to ablate macrophages. One thing that I failed to mention as well is that macrophages can be ablated in a temporal manner as well. They're not ablated until you give the diphtheria toxin. So you can wait until tumors form, for example, and then ablate the macrophages and see what happens. So here is my hypothesis, and that is that if we deplete macrophages in the mouse model of pancreatic cancer, we'll increase survival and we'll increase sensitivity to chemotherapy. That's the other thing that I'm going to look at. And here are the specific aims. So first, and here's a schema down here, I will take a number of mice, these mice that I talked about generating, and either treat them with diphtheria toxin or not, or treat them or not, and then see about tumor formation. See if there's a difference in tumor formation. And then I also will determine whether depletion of macrophages increases the sensitivity to gemcitabine. The olive paper that I mentioned before hints that altering tumor stroma can do that, so it will be interesting. As I mentioned before, these are broad brush strokes. Here's another way of looking at this. So this mouse that we're at work developing, at 100 days, this is when most of these mice form tumors. For the first part of the experiment, they'll either be given control or human diphtheria toxin, and then we'll look at mortality endpoint. We have a lot of experience with the KPC mice looking at these mortality endpoints, so this should be fairly straightforward, I hope. And we'll take a look and see if there's any difference. Concurrently, we'll take the same mice. All of them will be treated with gemcitabine, and then again, concurrently, they will either be treated with the diphtheria toxin or the saline control, and we will see whether or not there's a difference with regards to sensitivity to the gemcitabine. Of course, adjunctly with this, we will look at, we'll first of all look and see if macrophages are actually depleted, and we'll also look at the, how the stroma changes, how the tumor changes. There are many ways. This is a preliminary study funded by the seed grant, and so there are many, hopefully many future directions in which we can take this. A lot of them have to do with mechanism. So why, if they do actually enhance survival, why would they? So we'll look at how the cancer cells respond, whether there's any difference there, looking at things like apoptosis, for example, or other things, proliferation. how do the other stromal cell types react? Macrophages communicate with all of the other stromal cell types as well. And then we can look at specific cytokines which are elaborated by M2 macrophages and see if that has a role. And then if gemcitabine efficacy is enhanced as well, we can look at tumor levels of gemcitabine. Is it because the vasculature is more intact and more gemcitabine can get to the tumor? Or also hypoxia. Pancreatic cancers, I didn't mention this before, are very hypoxic, and this can play a role in pancreatic, or resistance to pancreatic cancer treatments as well. So with that, I would like to acknowledge, first of all, Chris Counter. He's my mentor. He's been a terrific mentor. The others in the lab, and then areas of funding, including the Hirschberg Foundation. So thank you.